Now that we've looked at passive and active transport through the plasma membrane, let's finish up talking about the plasma membrane, and then we'll proceed through the cytoplasm into the nucleus. So the plasma membrane does cell and environment interactions. For that reason, we need to talk a little bit more about the glycocalyx, because the glycocalyx is always involved when the cell interacts directly or even indirectly with other cells. Things that cells can respond to are things like extracellular chemicals, whether they be ions or molecules dissolved in a solvent, or even things like hormones or mucus or neurotransmitters. So here is a cell membrane. So we have our cell membrane, we have the blue hydrophilic heads, and then again in the middle, the phospholipid bilayer are hydrophobic tails. There are hydrophilic heads both facing the inside or intracellular environment of the cell and the extracellular environment of the cell. We have a couple of integral proteins embedded all the way through. No big peripheral proteins, but here's one, here's another one. Again, integral and peripheral proteins are different. We can see that we even have a membrane protein channel. Also attached to some of these proteins and even the phospholipid heads are glycoproteins or carbohydrate structures. So if they're attached to the phospholipid head, we call it a glycolipid. Whereas if it's attached to any of these proteins, they tend to be called glycoproteins. And that glyco part is coming from the sugars that make up these carbohydrate chains. We can even see in this light micrograph a tiny fuzzy layer outside the cell membrane. That fuzzy layer is the glycocalyx, which you can think of as a sugar coating. There's different types of glycocalyx molecules. We're going to talk about two large families, and those are the CAMs and the plasma membrane receptors. So the cell adhesion molecules are the CAMs. They're essentially molecular Velcro. They're things like cadherins and integrins. Desmosomes are going to use some of these proteins. And they're really going to act as arms or, again, molecular Velcro to help the cell hold on to other things, maybe other cells or other tissues. But they can also act as a distress signal if they get interrupted or partially destroyed. They're mechanical sensors, so they're going to be able to respond to fluid buildup on either side, but usually on the outside. And they're able to transmit intracellular signals. So they're going to function in signal transduction, and these direct signals generally are able to conduct cell migration, proliferation or copying of cells, and even specialization of cells as they grow. The plasma membrane receptors are, as you might expect, in the plasma membrane, and they're generally glycoproteins, so again, carbohydrate molecules bound to usually integral proteins, and they would function in contact and chemical signaling. They would have something bind to them, and what binds to the plasma membrane receptor is a ligand. And now if a plasma membrane receptor is acting as an enzyme, we could call that ligand a substrate, but they're essentially the same thing. And one of the most common pathways that plasma membrane receptors help facilitate is second messenger cycles or second messenger systems. A lot of amino acid hormones are going to use second messenger systems in order to get a signal from outside the cell to inside the cell without actually having something go through the cell membrane. Cyclic AMP is a second messenger for many amino acid hormones. So let's just talk briefly about a second messenger system. Essentially, a second messenger system starts with a first messenger, which is the ligand. The first messenger may be a hormone. We've talked about amino acid hormones in that last slide. And the hormone would bind to a receptor, which activates a G protein. The G protein would move to an enzyme, activate that enzyme, and cause that enzyme to create a second messenger by causing the enzyme to catalyze a reaction. Now the ligands, which are the chemicals that bind specifically to a plasma membrane receptor, again, I called it a hormone as a good example, but neurotransmitters are another example. They're going to be fundamental really to a lot of different chemical reactions that happen inside the cell. This process where we have a signal outside the cell and we're trying to get something to happen inside the cell without sending anything straight through the plasma membrane or even through a channel or through a carrier. That is called signal transduction. That's the transmission of molecular signals from a cell's exterior to its interior. And this is an amplification process. So we start with one first messenger, the ligand, that binds to its receptor, activates a G protein, which moves to an enzyme, which is the effector, and the effect it has is to break down or catalyze a reaction to create a second messenger. Here is the second messenger cyclic AMP or CAMP, and that's going to initiate a lot of different reactions by activating protein kinases. Protein kinases can create or turn on or turn off a lot of cellular responses. And generally the function of a ligand when it acts in a second messenger system is either to increase or decrease a process or maintain a process within the cell. Here's the picture you have in the book, figure 3.16. It just shows you the same thing. You can think of second messenger systems as running a relay race. So we'll start the relay race with our ligand, our first messenger, and we'll end it with our second messenger. Let's look at how complicated the extracellular environment is. So we will eventually talk about all the organelles, all the cell machinery you find inside cells. But cell signaling requires a lot of different proteins. This transduction, in order to get things to happen inside the cell, is also going to be a part of the extracellular environment. But now we're looking at the CAMs, the cell adhesion receptors. So you can see in this extracellular matrix, so this is outside the plasma membrane, we have plenty of carbohydrate and protein and kind of cytoskeletal elements that are helping hold this cell to its extracellular environment, but also helping stabilize the cell membrane in a particular location. Other roles of the membrane receptors, whether it's the CAMs or the cell membrane receptors, are contact signaling and chemical signaling. Contact signaling has to do with touching or the recognition of cells. Some immune cells perform contact signaling, and that's just a checkup on cells to make sure they haven't become infected with a virus and that they're supposed to be in the body. It's a way to identify targets that may be harmful. Then there's chemical signaling, which is an interaction between receptors and ligands to alter activity of cell proteins. So enzymes opening or closing or facilitating a chemical reaction, that would be an example of chemical signaling. Oftentimes, chemical signaling is shown in the second messenger systems because we're able to alter cell proteins inside the cell. So this is a dynamic of, again, that signal transduction. Alright, so that kind of ends our plasma membrane talk. We'll mention it. We can't forget about the plasma membrane. It's a vital part to the cell. The next part of our basic cell was the cytoplasm. So next, let's look at its composition and its structure and, of course, its function. Now, it says all about the cytoplasm, but it's really just the basics. First, what is the cytoplasm? Well, it's the fluid within the cell plus all of the cell machinery. We would call that fluid the cytosol, and it's a gel-like solution. It is mostly made up of water, but some soluble molecules that make it a little thicker. So it's basically water with solutes. It has some protein, salts, sugars, ions. But the big stuff that would be in the cytoplasm is the cytoplasmic organelles. And organelles, you should think of those as tiny organs for the cell, just like our organs help us carry out our day-to-day life. The organelles in the cell help them carry out their day-to-day activities. Oftentimes the metabolic machinery of the cell, the functions of the cell, are carried out by these organelles. There's also some inclusions, not in all cells, but these are things that don't dissolve in the water of cytosol. So they exist in just these areas within the cell because they're insoluble molecules, and they can vary with each type. Sometimes it's for storage of sugar, like glycogen granules in the liver. Other times it's pigment, like that we see in some cells that help create things like bile. And then there's lipid dropules in things like adipose cells. And even vacuoles help hold some water, or there's just crystalline structures from calcium buildup in some cells. So these inclusions are also mentioned, but are not the two main components of cytoplasm, which are cytosol and the cytoplasmic organelles. What we're going to look at next, because these organelles reside in the cytosol of the cytoplasm, are the membranous and non-membranous organelles. I do want you guys to read your textbook because it gives you a lot more information, and we're just going to hit the high points of each organelle and talk about ways to remember what they do, what is their function, because there's quite a few of them. So we'll start with the membranous and then move on to the non-membranous. And so we'll start with mitochondria, peroxisomes, lysosomes, endoplasmic reticulum, which actually has a smooth and rough variety, the Golgi apparatus, and then look at the cytoskeleton, centrioles, and the ribosomes. So first and foremost, you may have heard of mitochondria. Maybe in high school you might have heard powerhouse of the cell, but it's also known as the power plant of the cell. But both work because both for good reason are pivotal or right on the money when it comes to what mitochondria do. And that is they produce energy. They produce cellular energy and they produce cellular energy in the form of ATP. They're able to produce this ATP via a process called aerobic cellular respiration. That means that this process requires oxygen in order to occur. They're also interesting because they contain their own DNA and RNA, and so there's an endosymbiotic theory that suggests that at one time mitochondria were bacteria or early prokaryotic cell that were all on their own, and in order to get guaranteed nutrients, like the cells we have get, it went into an early eukaryotic cell, and it formed a symbiosis with the early eukaryotic cell and has never left. Next, we're going to look at ribosomes. Now, you can find ribosomes on the rough endoplasmic reticulum. That's why it's called rough endoplasmic reticulum, because the ribosomes give it a dotted, bumpy structure. But ribosomes, even if they're free and floating in the cytosol or bound to the rough endoplasmic reticulum, they are the site of protein synthesis. So that means part of the function of the rough endoplasmic reticulum must also be protein synthesis. So they allow for the formation or the anabolic reactions that make proteins. Next, we're going to look at the endoplasmic reticulum, which is usually located right outside the nuclear membrane. It is really going to be sacs of interconnected tubes. The smooth ER is a little bit more tubular in structure, where we have flattened sacs for the rough ER. And rough and smooth ER are a lot easier to say than rough endoplasmic reticulum and smooth endoplasmic reticulum. The biggest difference between the two in appearance is going to be that the smooth ER does not have ribosomes on it, whereas the rough ER does have ribosomes. So here is a histograph of the rough ER. So we're looking at the flattened sacs, we're looking at its external surface, we see the dotted or studded ribosomes, so rough endoplasmic reticulum. So it's named after its appearance. We know that ribosomes are the site of protein synthesis, so it should make sense that rough ER also has a role in protein synthesis, but it also can synthesize some other things, like some plasma membrane proteins, some integral proteins, but also those phospholipids. So it can synthesize proteins, even those that go in the plasma membrane, and then the phospholipids that make up the plasma membrane. So it's very much associated with not only the protein synthesis, but generation of parts of the plasma membrane. Smooth ER, it's important to note, has no role in protein synthesis. It is not embedded or studded with ribosomes, and it's much more tubular than flattened. What it functions in is varied. The most important ones, the ones that are generally mentioned, are things like lipid metabolism or detoxification. Detoxification is a big one, so liver cells or hepatocytes, for example, would have a lot of smooth ER because the liver cells are able to detoxify. Lipid metabolism is talking about cholesterol and steroid-based hormones, and their synthesis is carried out by the smooth ER in cells that need to produce such structures or molecules. But we're also going to see things like storage and release of calcium, converting glycogen to free glucose, which actually happens in the liver as well, like the detoxification, and then absorption, synthesis, and transport of fats. So things to do with lipids, detoxification, and storage, but no role in protein synthesis. All right, next let's look at the Golgi apparatus. It's great for transport, so I like to call it the post office of the cell. So it would actually take these budding vesicles that are filled with proteins from the rough ER. The vesicles would fuse with the Golgi apparatus, and as they travel through the green flattened sacs of the Golgi apparatus, these proteins would be modified or folded or concentrated and packaged, like what you see happens to your stuff at the post office, and then sent out to the right location. Maybe they're going to be sent to the cell membrane in order to be excreted. Secretion via exocytosis is one function of the stuff in the vesicles that comes from the Golgi apparatus. Or maybe the rough ER made phospholipids, so we're going to add those via vesicle to the plasma membrane. That way the cell can grow. Or maybe we need to digest some of these things that came from the rough ER. Or maybe there's stuff coming from the smooth ER, which can occur and still go to the Golgi apparatus. And then we would need to combine those things with a lysosome, create a phagosome, and digest what's inside that vesicle. So there's 3 different pathways that things can go as they process through the Golgi apparatus. And things are generally going to process from the rough ER out towards the Golgi, and then out further closer to the plasma membrane or to other organelles. The Golgi apparatus is generally depicted in green, although not always, but it's generally depicted in green, and it's a lot closer to the plasma membrane than the rough ER. So between the rough ER and the plasma membrane, you would find the Golgi apparatus. Next, let's talk more about the lysosomes. So we're going to have things called lysosomes and then things called peroxisomes. So kind of similar in their suffix or their ending. So remember that lysosomes are for digestion or degradation, whereas peroxisomes are going to be for detoxification. So lysosomes and peroxisomes are spherical membranous bags, but lysosomes contain digestive enzymes, usually acid hydrolases, and so they would take in substances, usually fuse with a vesicle and create a phagosome in order to digest either ingested bacteria, proteins, viruses, toxins. If something's going wrong with an organelle, it can ingest part of that and degrade it because it's non-functional. It also break down and release glycogen in the liver and skeletal muscle, and even break down bone to release calcium and phosphate. It also functions in cell autolysis. During cell autolysis, lysosomes bust and release their digestive enzymes into the cell in order to destroy the cell. We generally consider lysosomes a safe way to digest things because they hold all their digestive enzymes inside. So except for autolysis, they're going to be safe locations or safe sites because they isolate potentially harmful intracellular digestion from the rest of the cell. So peroxisomes are also membranous sacs, but they contain powerful oxidases and catalases as opposed to the acid hydrolases. They detoxify, not digest, so they'd be able to break something down into, say, water and oxygen. That's exactly what catalase does. Catalase breaks down hydrogen peroxide, which can be damaging to our DNA and cause point mutations, and breaks down hydrogen peroxide into water and oxygen. So it's definitely able to neutralize things, but really break them down into smaller particles. It just makes sure that they're no longer harmful. Alright, next we're going to be looking at more organelles, but what we've been looking at we call the endomembrane system because it was membranous organelles that we were looking at. They had an inside and an outside, they had compartments. So the rough and smooth ER, the Golgi apparatus, secretory vesicles, so that includes lysosomes, all of these were membranous organelles. And these were able to produce something, break down something, store something, export something, depending on which membranous organelle you're looking at. They were even, if we're talking about peroxisomes, able to degrade potentially harmful substances by detoxifying or neutralizing them. So next we're going to be looking at non-membranous organelles, and that begins with the cytoskeleton. This is really going to be kind of like protein strings that you find within the cytosol, within that fluid. They don't create compartments, but they do provide a lot of support for the cell. So there is an elaborate series of rods throughout the cytosol. Collectively, we would talk about these three things, the microtubules, the intermediate filaments, and the microfilaments, as the cytoskeleton. And really take that word exactly as it means, which is that we're looking at a cell skeleton, because these are providing that structure, that support to the cell. So we'll start with microfilaments. Microfilaments are the thinnest of the cytoskeletal elements. They are made up of actin subunits, which are these circular subunits that link together. Usually two strands link together and then wrap around each other to create an actin strand. And these strands, which are made of spherical protein units, again, those are the ones called actin, are really able to give the cell motility. If a cell is able to produce pseudopods or move from an area to another, such as an amoeba, which is not a cell that's in the human body, but if it's able to produce those pseudopods, it's because of actin. We can also see that a cell changes shape or responds to stressors in its environment, can perform endocytosis or exocytosis because of the actions of actin. Next, there's the intermediate filaments, which are intermediate in size between the microtubules and microfilaments. And the intermediate filaments are definitely tough and rope-light. They're not made of actin, they're made of fiber subunits. So hence them being called intermediate filaments. They really are just that. They are going to function in desmosomes because they resist pulling forces on the cell. They're attached to those desmosomes, so they function mainly in that particular cell junction. Microtubules are probably the most interesting. They're the largest of our cytoskeletal elements. they're made up of tubulin, so microtubules are made up of tubulin, microfilaments are made up of actin as far as subunits are concerned. So they're very dynamic, they form rope-like structures, but they're hollow tubes. They tend to radiate from another organelle called the centrosome, or called the centriole, which exists in the centrosome. So centrioles are the organelle, and centrosomes the area where the centrioles hang out, and that's why the microtubules radiate from there, because the centrioles create them. And they determine the overall shape of the cell and distribution, but they're mainly good for cell-to-cell trafficking. They're going to work with motor proteins in order to move secretory vesicles from one area of a cell to another, or move mitochondria or a different organelle from one area to another. Motor molecules, we don't have a picture, but things like dynein is a good example. There are protein complexes that actually sometimes seem to walk on things like microtubules, kind of using microtubules as a cellular highway. They do need energy, so they're powered by active transport or powered by ATP, and they help proteins move from one area to another, or organelles move from an area to another. Usually proteins are moving inside vesicles, so the motor molecules act like train engines. So I mentioned the centrosome and centrioles. These centrioles are organelles, and these organelles produce microtubules. The area in the cell where you can find the centrioles, usually a pair of centrioles, is called the centrosome. So again, centrosome is an area, usually close to the cell center or the nucleus, and the centrioles are the organelle. You generally have a pair, and they produce microtubules. They are also the basis for the structure that produces cilia and flagella. Cilia are short compared to flagellum. Cilia tend to be on the cell surface. They whip back and forth, and they would move things past the cell, kind of move fluids along the cell surface, whereas the flagellum, being much longer, is able to propel whole cells, like spermatozoa or sperm. Microvilli are different from cilia and flagella, but all three are cellular extensions. But cilia sit on top of the cell. Flagellum is going to extend from the cell, and they are true cellular extensions in that they are structures on or embedded in the cell. Microvilli look like cilia. They're hair-like extensions, but they're actually extensions of the plasma membrane itself. So if we went down here to the plasma membrane, it's stretched up and then down, up, down, and that just kept going. So it's very different from cilia and flagellum in that they're not separate structures embedded or attached to the cell. It's the actual plasma membrane that has formed these hair-like structures. And finally, we're going to talk about the nucleus. So the nucleus is where DNA resides, so it has the genetic library with the blueprints for the instructions for how to make cellular proteins. So DNA is found in the nucleus. The nucleus does respond to the outside environment. It responds to what's going on in the cytosol. So it responds to signals, it dictates different kinds and amounts of proteins to be synthesized. And most cells just have one nucleus, which is called being uninucleate. If you have more than one, if it's just two, sometimes that's binucleate, but multinucleate is more commonly used, which is more than one nucleus. And some cells, very early on in their development, get rid of their nucleus, and the red blood cell is an example. So red blood cells are anucleate, meaning they do not have a nucleus. There's three basic regions to the nucleus. You have the nuclear pores, nuclear envelope, and the nucleoli. The nucleoli, sometimes there's more, but generally there's just one in eukaryotic cells, are going to be an area where rRNA is made. And rRNA is involved in the creation of ribosomes, and ribosomes create protein. So if you mess with the nucleoli, you mess with that nucleolus, and you mess with the production of rRNA, you can inhibit ribosome production and therefore inhibit protein synthesis. The nuclear envelope is double layered, it surrounds the nucleus, and it has pores that allow things in and out. DNA is never allowed out. It stays in the nucleus in the unwound form called chromatin. Chromatin is thread-like. It's just strands of DNA, usually mixed in a little bit with histone proteins, sometimes a little bit of RNA, that's usually mRNA or rRNA, that's already hanging out in the nucleus. We would call this combination of DNA and protein a nucleosome. And histones are able to pack long DNA molecules, and when we turn chromatin into chromosomes, these histones become really important for supercoiling DNA. So chromatin structure is unwound. Usually it exists as slightly coiled, where you have DNA wrapped around histone protein, so it kind of looks like beads on a string. And then when it starts to coil tighter and then coils upon itself and then coils even more and becomes really dense, that's when we're starting to form a chromosome. So chromatin, you can think of that as unwound DNA in the form of histones wrapped around the DNA double helix. Whereas chromosomes, you can think of that really compact supercoiled DNA, and those compact supercoiled DNA structures are easier to move than the unwound DNA. So that brings us to why we would want really compact DNA, and that's in order to move it around. In order for cell division to occur, in order for division of the nucleus, therefore movement of the genetic material to occur, you have to have it in kind of movable pieces. So we're going to talk now about the cell cycle. The cell cycle is just a series of changes that a cell undergoes from the time it is formed until it reproduces. So essentially, a cell's life cycle is the cell cycle. It includes two major phases, interphase and cell division. Cell division is also called mitosis. So interphase and mitosis, you could also write that as the two major periods of the cell cycle. Interphase is when a cell is carrying out its usual activities, carrying out its day-to-day life, performing metabolism, making energy, whereas cell division divides the cell into two, but specifically divides the nucleus in two. So the best way to define, really, mitosis would be nuclear division or division of the nucleus. So cell division, cell cycle, we had two phases, interphase and mitosis. You can see that most of the time a cell would be in interphase, and only for a short period of time would it divide and be performing mitosis. The four steps of mitosis are prophase, metaphase, anaphase, and telophase. A process called cytokinesis occurs during telophase. So we'll come back to all that and look at that in depth. But let's talk about G1, S, and G2, which are the three subphases of interphase. G1 and G2 have checkpoints where they check that either growth signals or the integrity of cellular DNA is fine, or make sure nothing has been ruined and the cell's ready for mitosis, is ready for cell division. The G1 checkpoint checks for growth signals being prepared properly and that DNA hasn't mutated. The G2 checkpoint checks to make sure that the DNA was copied properly. And DNA is copied during S phase, or the synthesis phase of interphase. So the S phase of interphase is when DNA is replicated. So during interphase, not a lot's going on. Cell-to-cell signaling could be occurring, we could be making ATP, but as far as anything different from day-to-day life for that cell, not really occurring. You should see two centrosomes. Normally, right before a cell replicates itself or copies itself during mitosis, it copies its centrosome. So it'll have two areas, both with a pair of centrioles. And remember, the centrioles are going to produce microtubules. Plasma membrane will be intact, nuclear membrane is intact, nucleolus is present, and DNA exists as chromatin. And again, that's what a cell looks like in interphase. And so it's a period before mitosis. It's a period of regular cell function. It's a period during which DNA exists as chromatin. And it consists of those three subphases, one of which is S phase, during which DNA replication occurs. And DNA replication in eukaryotic human cells is semi-conservative. And what this means is that our DNA molecules during S phase split, and each strand of the double-stranded DNA gets a new strand. So each new strand of replicated DNA would have an old and a new strand. So that means that each nucleotide strand is going to serve as a template for building a new complementary strand So we'll get two DNA molecules formed from the original, but again, semi-conservative, because again, the replicated DNA, each piece would have a new and old strand. So let's look at that. So it is semi-conservative replication that is seen during the S phase of interphase. Here is the parent strand. If the parent strand splits and you have one old, another old, and we use the old strands, the red strands, as the template to create the new strands, then the two new strands produced from the parent strands are going to have an old and a new strand. But both of them do, so this replication was semi-conservative. We conserved part of the old or parent strand. I do recommend watching this video when it comes up in mastering. It's just going over replication of DNA. If it's no longer in your mastering homework, check out that study area that you have access to. If you have access to mastering, the study area has practice quizzes, videos, true false, labeling, tons of information that's good to be going over before you take online exams. Alright, in your PowerPoint you also have the subphases listed. So again, the subphases of interphase are G1, S, and G2. Synthesis or synthetic is what S stands for. And again, during S phase of interphase, DNA replication occurs. If you're catching on that that's the most important for the subphases, then you're doing well. Next we have mitosis, and mitosis is not a part of interphase. So again, the two processes of the cell cycle were interphase and mitosis. So we're moving to looking at mitosis now, and the definition of mitosis is division of the nucleus. Cytokinesis is a process that happens during the end of mitosis, and it would be division of the cytoplasm. So if we can divide the nucleus and the cytoplasm, we can divide the cells, and we create two new daughter cells. There are 4 stages in mitosis: prophase, metaphase, anaphase, and telophase. And telophase is during which cytokinesis begins and occurs and creates a cleavage furrow. So during prophase, there's a couple of things that occur. At the very end of prophase, the nuclear envelope breaks up, but first and foremost, our chromatin becomes chromosomes. So we can start to see chromosomes or start to see DNA form in the unbroken nuclear envelope. And as the chromosomes form or the DNA condenses, the centrosomes will migrate to opposite poles of the cell, and they will contain centrioles in them, and they'll begin synthesizing microtubules. During mitosis, we call these microtubules produced from the centrioles in their centrosome regions the mitotic spindle or spindle fibers. And then only at the end of prophase, once we have chromosomes, once we have the migrated centrioles in their centrosomes, will the nuclear envelope break up, and now we're ready to split that DNA that has been doubled before prophase. So now we're starting to see things happen. This is early prophase, so the nuclear envelope may begin to fragment here, but it occurs completely by the end of prophase. But you'll start to see the two centrosomes with their centrioles migrate to opposite sides and create the early mitotic spindle of spindle fibers. During late prophase, the nuclear envelope will be almost completely gone. It'll be completely fragmented or degraded, whichever way you want to think about it. The centrosomes will be on opposite poles, and the spindle fibers will be a little bit longer. They may even start to attach to the chromosomes. During metaphase, all of the chromosomes line up on the middle. So it's important to know what's lining up in the middle, and it's the chromosomes. So the chromosomes line up in the middle, they're attached to the spindle fibers, and your centrosomes with their centrioles are still on opposite poles. So about midway between the two centrosomes would be the metaphase plate. So this is the M plate, or you can call it the metaphase plate, or you can even call it the equator of the cell. But we get all those chromosomes lining up in the middle. Both metaphase and middle start with M, and so that's what we're going to focus on for remembering what happens during metaphase. Both apart and anaphase start with A, and during anaphase, the sister chromatids are going to be pulled apart. So our X-shaped chromosomes will be pulled apart, and what's left is a sister chromatid, which was paired with the other chromatid, hence them being called sisters, and they're being separated now and being pulled to opposite poles of the cell as the mitotic spindle fibers shorten. And then during telophase, we see the opposite of prophase. We're going to see the chromosomes stop moving. We're going to see the chromosomes uncoil, so they'll become chromatin again. We're going to see the nuclear membrane come back, and we're going to see the spindle fibers disappear. So during prophase, the spindle fibers appeared, the nuclear membrane went away, our DNA formed chromosomes, and just the complete opposite happens during telophase. The only thing that's not the complete opposite is cytokinesis. During telophase, cytokinesis also begins. Cytokinesis is not an extra phase of mitosis. It's just a process that occurs at the end of mitosis. And cytokinesis, which occurs mainly during telophase, actually begins as soon as the chromosomes stop moving, which is in anaphase technically, and then we move to telophase. And cytokinesis is the division of the cytoplasm. The way that you would see this division occur in animal cells like those of the human body is via the production of a cleavage furrow. So this cleavage furrow is produced via a ring of actin microfilaments that form around the middle of this elongated cell and start to cleave or contract and separate the cytoplasm. So during telophase is mainly when cytokinesis occurs. You start to see these divots or these indents on the side of the cell. And if you're looking through a light microscope, that's the giveaway that you're looking at telophase and not anaphase. Because even though your textbook says cytokinesis starts in anaphase, it starts at the very end. So if you see this in a light microscope, you're looking at telophase. Alright, this is also an animation that should be available to you on Mastering. Alright, and then one of our last topics is going to be over the central dogma of molecular biology. And this is just a fancy term to say that we're going to use DNA to make protein. And in order to read DNA and get that signal from the nucleus out into the cytoplasm to a ribosome, we're going to use RNA as our intermediary molecule. So DNA is definitely the master blueprint. It's our blueprint for reading how to make a protein. And mRNA is able to read that blueprint and bring it to a ribosome. And then mRNA and the ribosome work together with tRNA, which is transfer RNA, in order to make a protein. The part of DNA that mRNA would read is called a gene. And you read genes, which are pieces of DNA in triplets. We call them DNA triplets. They're just 3 sequential nitrogen-containing bases that are basically forming separate nucleotides. So we're going to talk briefly about nucleotides. If you don't remember, nucleotides make up nucleic acids. Nucleotides are made up of nitrogenous bases, a phosphate group, and a sugar, usually a pentose sugar. For DNA, the bases are adenine, guanine, thymine, and cytosine. We saw uracil in RNA instead of thymine. And each DNA triplet is going to be specific for an amino acid. And amino acids, if you put them together, just like if you put nucleotides together, you get DNA. If you put amino acids together, you get a protein. So we have all these components that are necessary for bringing stuff together in order to make a certain biological molecule. In this case, we have all the components we need in order to follow a blueprint, bring the amino acids and the machinery together to build a protein. So let's talk about protein synthesis. Once you make the protein, there's good parts and bad parts. There's exons and introns. The exons actually code for amino acids, and then the introns are non-coding segments. So you'd actually cut those out of the finished protein product. RNA, again, is our intermediary. We have 3 different types of RNA. mRNA is going to be the big one. It's messenger RNA. It's the messenger from the nucleus to the cytoplasm, talks to the ribosome. You also have ribosomal RNA made in the nucleolus of the nucleus. That's going to help build the ribosome. And then transfer RNA is going to bring amino acids to the ribosome once we're reading mRNA. Now, RNA differs from DNA again. Uracil is going to be substituted for thymine, and RNA is single-stranded, whereas DNA is double-stranded. Again, RNA is the intermediary, so it's going to link DNA to proteins. So that's why we're seeing it in that central dogma of molecular biology. We're seeing it between DNA and protein in that flowchart. All RNA is formed in the nucleus, whether it's mRNA, rRNA, or transfer RNA. Again, mRNA is messenger RNA, rRNA is ribosomal RNA, and transfer RNA is for tRNA. RNA also has a ribose sugar instead of a deoxyribose sugar, so that should be it for our recap on differences between DNA and RNA. If you also want to know the roles of the three main types of RNA, you have them here. Messenger RNA is a messenger. It's going to bring the instructions from DNA, from reading a gene, to ribosomes in the cytoplasm. rRNA is a structural component of ribosomes, hence it being called rRNA, which stands for ribosomal RNA, and it's going to work along with tRNA to translate the information brought to the ribosome by or from mRNA. And then transfer RNA is going to bind to amino acids and bring them to the ribosome in order to put the amino acids in the right order based on the reading of mRNA. Now there are two processes for creating a protein. So the first step, we have to go from DNA to RNA. We have to convert our DNA blueprint into an RNA message. To do that, it's called transcription, because they're both going to be nucleic acids. So if you're taking notes, and I'm speaking in English, not necessarily, but most of the time, you'll be writing notes in the same language that I'm speaking. So you will transcribe notes in a similar manner to what I'm saying. And so that's exactly what happens during transcription. We have a nucleic acid DNA, and we're going to turn it into another nucleic acid, mRNA. Maybe there's some shorthand, there's some slight changes like uracil instead of thymine, but they're still both nucleic acids. So that first process where we go from DNA to RNA is transcription. Now, if we change the language, if we change the molecular language, then it's translation. So if we go from a nucleic acid RNA to building a protein, those are two separate types of biological molecules. So that's translation. To go from nucleic acid, mRNA, to protein or polypeptide, that's translation. So again, DNA to RNA, that process is transcription. RNA to protein, that's translation. And here's a great image to study, because it shows you that transcription must occur in the nucleus, where mRNA is made. So DNA to mRNA is going to be transcription, and we have to get rid of those introns, which are kind of our junk mRNA. So there will be some RNA processing before we have our mRNA that actually leaves through a nuclear pore. The mRNA will then go to a ribosome and kind of gets fed through like a ribbon that you see in this image, and it gets read. And thanks to the rRNA, which helps build the ribosome, and the tRNA, which brings the appropriate amino acids, by reading the mRNA coded instructions, we can build a polypeptide or a protein by putting those amino acids together. So transcription transfers DNA base sequences to a complementary base sequence of an mRNA. Said simpler, we read the blueprint of DNA, make an mRNA transcript, and send that out of the nucleus to talk to a ribosome. There's different things like transcription factors and gene activators. We have to loosen part of the histones, that's protein wrapped around DNA, in order to really get at that genetic blueprint. There's promoters, initiators, there's mediating enzymes like RNA polymerase and DNA polymerase. But we just want to know the base idea behind transcription. Yes, there's enzymes involved, like RNA polymerase, which is going to oversee the synthesis of mRNA. Makes sense. RNA polymerase. It's going to make a polymer, which means something with many, of RNA. So it's going to make a type of RNA, and it makes mRNA. It has ACE at the end, so that tells us it's an enzyme. A lot of things can be drawn just by the names of things in biology. But we want to know, again, transcription is the formation of mRNA from a DNA blueprint. We read one gene to make one mRNA transcript, and transcription occurs in the nucleus. Translation, if we read what's here in red, converts base sequences of nucleic acids into an amino acid sequence of protein. So by the time we get to translation, our nucleic acid is mRNA. We're going to use tRNA and rRNA in order to make the protein. But we could say this really simply to say we're going to turn mRNA into protein or polypeptide during translation. We're going to change the molecular language. But it's going to use all three types of RNA. Now, if we were to look at a codon chart, which is what's on the right here, we would be looking at it in order to read the genetic code. So what's on the codon chart is going to be nucleotides, but they're specifically mRNA nucleotides. And the sequence of 3 mRNA nucleotides is called a codon versus a DNA triplet. And in order to read mRNA through a ribosome, we read codons, or I should say the ribosome reads codons, and then tRNA would bring the right amino acid. For example, UUA as a codon would bring leucine. And you can see that there's a couple of different codons for each amino acid, and there's 20 amino acids total. So during translation, mRNA attaches to the small ribosomal subunit. There's a small and a large. Then the large ribosomal subunit attaches. Now you have a functioning ribosome. We're going to send mRNA through the ribosome, and whereas mRNA has codons, tRNA has anticodons, and the two match up if they recognize each other, and that's how the correct amino acid is added to a growing polypeptide chain as this anabolic reaction occurs at the ribosome. At the end, you will have created a protein. So to summarize, we're going to use complementary base pairing. That way our anticodons and codons pair up. And the central dogma of molecular biology just states that we take DNA triplets and turn them into mRNA codons. The mRNA codons are read at a ribosome in order to bring the right amino acid, and we will build a protein from those amino acids carried to the ribosome by tRNA. And then rRNA, of course, makes up the ribosome. And there's other roles of DNA. DNA codes for other types of RNA besides mRNA. There's microRNA, riboswitches, and small interfering RNAs. Some of this stuff is really new. For example, small RNAs that can bind to and silence mRNAs made by certain exons, that's microRNA. So there's some editing functions of RNA on DNA. Riboswitches are similar. They're folded pieces of RNA that can also turn on or off protein synthesis, depending on certain environmental conditions, whether or not a cell really needs that protein. So these are basically gene on-off switches. Even small interfering RNAs, which are abbreviated siRNA, are very similar to miRNA or the microRNA, and they can silence mRNA, which would prevent the generation of certain proteins. So they're basically gene on-off switches, whether they do that at the level of DNA or mRNA or even at the ribosome, depends on the type. And that's it for Part B, so make sure to go back and look at Chapter 3, Part A, and then again, Chapter 3, Part B, 1 and 2.